Hello, class. Today we're going on to chapter 10, where we talk about senses. The objectives are to classify sense organs as general or special and explain the basic differences between the two groups. Discuss how a stimulus is converted into a sensation. Discuss the general sense organs and their functions. Describe the structure of the eye and functions of its components. Discuss the anatomy of the ear and its sensory function in hearing and equilibrium. Describe the anatomy of the tongue and its sensory function in taste. Describe the anatomy of the nasal cavity and its sensory function in smell, and discuss how senses are integrated. So, classifications. We have general senses and special senses. So, general senses often exist as individual cells or receptor units, widely distributed throughout the body. Special senses are large and complex organs. These are a localized grouping of specialized receptors, and this includes smell, taste, hearing, and equilibrium. Then we have something called sensory receptor types. This is classification by presence or absence of covering capsule. We have encapsulated, and then we have unencapsulated, which are also known as free or naked nerve endings. So, classification by type of stimuli required to activate receptors. So, we have photoreceptors, chemoreceptors, pain receptors, thermoreceptors, mechanoreceptors, proprioceptors. So, let's go into each of these. Photoreceptors, basically, this is how we are able to sense light. This occurs in our vision through rods and cones. Chemoreceptors are chemicals. These are part of smell and taste from the nose, which is called olfactory cells, and the tongue or taste buds that are called gustatory cells. We have pain receptors in response to injury, and these come from free nerve endings in the skin and mucosa. They're not evenly distributed throughout the body, and they are quite microscopic. Then we have thermoreceptors. These sense temperature change, again, from free nerve endings. Mechanoreceptors, these sense movement or deforming of the capsule. These are found in the ear, which the organ of Corti is the actual organ that we use in hearing. And then we have dynamic and static equilibrium or balance. Then we have proprioceptors. These are in response to a position of body parts or changes in muscle length or tension. If you don't have any proprioception or you have poor proprioceptors, you're also going to probably be a little clumsy. The tendon organ, the Golgi tendon organ, is the organ of proprioception. Now, all sense organs have common functional characteristics. They are all able to detect a particular stimulus. A stimulus is converted into a nerve impulse. The nerve impulse is perceived as a sensation in the central nervous system. The sensory pathway for general senses typically involves conduction of action potentials generated in the receptors through the spinal cord to the thalamus. The sensory pathways for special senses are varied, but they also ultimately end in a specific sensory areas of the cerebral cortex. Now, general senses. These are microscopic general sense organ receptors that are found in almost every body part and organ. The most are concentrated on the skin, but they are not evenly distributed. Distribution is widespread. They are single cell receptors that are common, and examples include free nerve endings in the skin and mucosa that sense pain, temperature, crude touch. Then you have tactile or Messner corpuscles in the skin, fingertips, and lips. These sense fine touch or vibration. Then we have bulbous or Ruffini corpuscles, and this is in skin and subcutaneous tissue of the fingers. These sense touch and pressure. Some examples continued that we have the lamellar or Pacini corpuscles that we talked about before when we talked about the skin. This is in the subcu or subcutaneous, submucosa, around the joints, and the external genitals. These sense pressure and vibration. The bulboid corpuscles, which are known as Krausen bulbs, this is in the dermal layer of the skin, the subcutaneous, mucosa of the lips, eyelids, and external genitals, and they sense touch. Then we have our Golgi tendon receptors. These are near tendons and muscles, and it's very important for proprioception. So Golgi tendon receptors are important with proprioceptors. We have muscle spindles, skeletal muscles, and they're also important for proprioceptors. Now, the ability to distinguish one touch stimulus from two is called two-point discrimination. So this gives you an idea of general sense receptors. There's free nerve endings, little teeny tiny ones you see up in the epidermis. And then in the dermis, you'll see the balboid or the coarse corpuscle, the tactile Messner corpuscle, lamellar Pacini corpuscle, and the bulbous Ruffini corpuscles. So in the previous slides, I talked about what they do, like fine touch, vibration, things like that. You have to know those. Now let's move into special senses. We're going to begin with vision. This is the structure and function of the eye. So we have the sclera, which is the fibrous, tough outer coat or white of the eye. The cornea is the transparent part of the sclera that's over the iris. The cornea is the transparent circle on the anterior of the fibrous layer, or the front, and it is the window of the eye. When inflammation of the cornea happens, we call this keratitis. Then you have your conjunctiva. This is the mucous membrane covering the front surface of the sclera and lines the eyelid, and it is kept moist by tears found in the lacrimal gland. Moving on, we have the choroid. This is the pigmented vascular layer that prevents scattering of light. It is the front part of this layer that's made of ciliary muscle and iris, the colored part of the eye, and the pupil is the hole in the center of the iris. Contraction of the iris muscle dilates or constricts the pupil. The iris is the colored structure seen through the cornea. The pupil is the black center of the iris. It's donut shaped and it contracts in bright light and dilates when letting more light in. The lens is the transparent body behind the pupil. It focuses light rays on the retina. And the retina is the innermost layer of the eye. It contains rods, which are receptors for night vision, and cones, which are receptors for day vision and color vision. There is a yellowish area near the retinal center called the macula lutea. It surrounds a slight depression that we call the fovea centralis, which you need to know. This contains the greatest concentration of cones of any area of the retina. So here you can see all of those areas that we just talked about. And I do recommend that you study this to get an idea of where they're located in the eye. If you'll notice, there's only 3 layers of the eye. We have the inner layer, the vascular layer, and the fibrous layer. We're going to go on and talk more about the posterior and anterior chambers that have, anterior chamber has aqueous humor, which is a fluid, and the posterior chamber has the vitreous humor. So you can see where at the bottom the optic nerve comes into the disc, and you can see where all of these things work together to form the eye. Now, in the retina, when we talk about cones and rods, you can see on this image the difference, right? And make sure you know what rods do, what cones do. So let's move on to those fluids of the eyeball. The aqueous humor is in the anterior chamber in the front of the lens. The vitreous humor is in the posterior chamber behind the lens. Now, if drainage of these fluids is blocked, the eye's internal pressure increases and damage could lead to blindness. This is called glaucoma. The visual pathway, this is the innermost layer of the retina that contains rods and cones. Impulse travels from the rods and cones through the bipolar and ganglionic layers of the retina, which is seen in image 10-4. Nerve impulse leaves the eye through the optic nerve. The exit point is free of receptors and is therefore called a blind spot. The blind spot is also known as the optic disc. Visual interpretation occurs in the visual cortex of the cerebrum, and the visual interpretation of the nervous impulses generated by light striking the retina results in what we call seeing. As we age, most people become farsighted as they lose the ability to focus on close objects. This is called presbyopia, or also known as old-sightedness. Now, exposure to UV light can cause the lens to become hard, lose its transparency, become cloudy, or milky in appearance, and we call these cataracts. Moving on to the ear, we're going to talk about hearing and equilibrium. The ear functions in hearing and in equilibrium and balance. Receptors are called mechanoreceptors, and the ear also functions as the sense organ of balance. The stimulation or trigger that activates receptors involved with hearing and equilibrium is mechanical. Therefore, it's a mechanoreceptor. So the external ear consists of the auricle, which is the pinna. Then we have, and that's, your earlobe and what we see. Then you have the external auditory canal. This curving canal is about 1 inch in length. It contains ceruminous glands. That's why we have earwax. And it ends at the tympanic membrane on the inside of the ear. As we go from the external ear, we next go into the middle ear. This houses the ear's ossicles. These are ear bones. The ear bones consist of the malleus, the incus, and the stapes. It ends in the oval window, which separates the middle ear from the inner ear. The auditory or eustachian tube connects the middle ear to the throat. So that's how everything drains into the back of the throat. Inflammation of the middle ear is called otitis media, or an ear infection. So you can see here, there's the external ear. You can see the auricle and pinna on the outside. You see the external acoustic meatus go in, and then we see the tympanic membrane. The tympanic membrane ends at the middle ear, and this is where we have those bones, the malleus, incus, and stapes that kind of move and vibrate with things coming in, vibrations of sounds. You can see at the bottom, this is the eustachian tube or the auditory tube that goes down. And then we're going into the inner ear. So the inner ear consists of 3 spaces that are located in the temporal bone. We have the bony labyrinth, which is a complex maze that's filled with perilymph. Perilymph is a thinner watery substance. It is subdivided into the vestibule, semicircular canals, and the cochlea. The membranous labyrinth is filled with a thicker fluid called endolymph. And that's important to know the difference between the fluids in the bony labyrinth and in the membranous labyrinth. The receptors for balance in the semicircular canals are called cristae ampullaris. And then there's specialized hair cells on the organ of Corti. Remember, the organ of Corti is the organ of hearing, right? That's why we can hear. So there's specialized little hair cells on the organ of Corti that respond with bent by the movement of surrounding endolymph set in motion by sound waves. This spiral organ, or the organ of Corti, is the organ of hearing. So this shows up close to the inner ear where you can see the vestibular nerve, cochlear nerve. You see the cochlea, you see the membranous labyrinth, bony labyrinth, oval window. So it's very, very complicated in here. You can see the sensory hair cells that move, and then you see right in here is that spiral organ of Corti, and that is why we can actually hear and discriminate sounds. So this shows the effect of sound waves in the ear. So sound enters the outside, goes into the external acoustic canal through the tympanic membrane. The tympanic membrane, when a doctor looks in our ears, it should be pearly gray, okay? If it's red or inflamed, that means that we have an ear infection. Something's going on. So normally, if the tympanic membrane's normal, pearly gray, not full of fluid, the sound will come out. It moves these bones, the malleus, incus, and stapes, depending on what the pitch is, goes into that endolymph, goes down, crosses over, down into the perilymph. These little hairs on that spiral organ of Corti pick up and sense the vibration and gives us the tone that we are hearing. The cochlear nerve penetrates the brain so that we understand what we're hearing. Now, equilibrium also occurs in the ear. The static equilibrium is the sense of gravity. It is detected by ciliated hair cells of the two maculae in the vestibule. When the head tilts, gravity pulls the heavy gel of each macula, bending the cilia and producing a nerve signal. That's why we don't fall over when we start moving our heads. Dynamic equilibrium is the sense of speed and direction of movement. This is detected by ciliated hairs of the crista ampullaris. When the speed or direction of movement of the head changes, the flow of endolymph in the semicircular canals is altered. The vestibular nerve carries nerve impulses from the equilibrium receptors, which are mechanoreceptors of the vestibule, joins with the cochlear nerve to form the vestibulocochlear nerve, and it goes into the brain. Now let's talk about taste. Remember, taste is a special sense, just like vision and hearing. So receptors are chemoreceptors called taste buds. Our sense of taste, or gustation, allows us to chemically analyze food before we bite or swallow it. Taste buds are made of supporting cells and chemoreceptors called gustatory cells. There are two cranial nerves, 7 and 9, that carry gustatory impulses. Most pathologists list 4 kinds of primary taste sensations. So we have 4 primary taste buds, right, or sensations. These are sweet, sour, bitter, and salty. However, there are 2 other unique tastes that we can pick up with our tongue, and this is metallic and umami. Umami is why we can taste meat, meaty taste. They are unique and may soon be added to the list of primary taste sensations, however, they have not been already. Now, nasal congestion can interfere with the stimulation of the olfactory receptors and thereby dull taste sensations. So smell and taste kind of go together. If you can't smell, you're not going to really be able to taste. So again, gustatory, which is the taste, and olfactory, which is the smell senses, work together to permit creation of many other taste sensations. Now here's a close-up image of the tongue. And you can see on either side of the tongue is the palatine tonsils. And at the back, you see the lingual tonsils that are kind of underneath the tongue there. You can see this papillae, filiform papillae. And so you see all of these little things. Well, what you don't see is under these little areas on the tongue are these taste buds. These taste buds are the gustatory cells. They have little hairs that put the taste down into the cell. And it basically, the tongue gives us our taste buds, right? Without the tongue, we're not going to have the taste buds. Now let's talk about smell. There are receptors for fibers of olfactory or cranial nerves that lie in the olfactory mucosa of the nasal cavity. The olfactory receptors, or smell receptors, are extremely sensitive but easily adapt and become fatigued. That's why if you smell something for a while, you can't smell anything else, right? That's why we smell coffee beans in between smelling perfumes and incense and stuff. Odor-causing chemicals initiate a nervous system signal that is interpreted as a specific odor by the brain. That's how we can determine that, I smell coffee or I smell flowers or whatever. The brain associates the smells, the sensations with certain smells. So this gives you an idea of the olfactory structure. So you smell it, it goes up and through the nasal cavity into the olfactory epithelium, through the olfactory bud to the cranial nerve into the thalamus. And there's a lot of other stuff along the way, but that's basically what's going on when we smell. Or even taste, because remember, the mouth is connected to the back of the throat, which is open to the nasal cavity. So smell and taste go together. Sensations are all perceived in the brain, not an individual receptor scattered throughout the body. Severe nasal congestion can interfere with the stimulation of olfactory receptors by food odors in the mouth, which may lead to dull flavor sensations. Smell sensations are often powerful triggers of memory due to the connection to the limbic system, the emotional area of the brain. So that is why smells bring back memories. And that is the end of chapter 10. So be sure to study, make sure you know all of the areas that I discussed of the eye and the ear and what they do. And pay attention to that. Again, you will not be labeling, but you do have to know what each one of them do. For example, what the pupil does, what the iris does, what the tympanic membrane should look like. It should be pearly gray, things of that nature. So get to studying, and I will be talking to you in the next one.